Hello students, this is Mr. Bishop, and we are starting off our semester with our first screencast, and we are going to do this one a little different. In class, and there is going to be a new level of interactivity to them. We'll work with the interactive app in class so you guys can work out any kinks you have working in EduCanon. So let's begin to find out what we and other living things look like under a microscope. The cell. As we discussed in our Intro to Bio chapter, the cell is the basic unit of life. All things that are living have one or more cells. I asked in class, what does the cell look like? We had some mixed answers, but here to the right we have an illustration of a cell. So this is what cells look like. Some of them, not all of them, but definitely some of them. So how do we see cells? Can we see cells with our eyes, or do we need tools to see a cell? The answer, of course, is yes. We need tools to see cells. We need microscopes. Microscopes are an amazing scientific tool. In a similar fashion that telescopes have enabled us to see objects far away in the universe, microscopes have enabled us to see objects so small the human eye cannot see here on our own planet. This enabled scientists to study bacteria. Prior to this, diseases were thought to be caused by supernatural forces. Anton von Leeuwenhoek came along and developed the first light microscope and said, no supernatural forces. Cells, I see cells. Using microscopes, we can see red blood cells and blood vessels. As I said before, with our illustration of our cell, we have more cells to the left in the shape of a heart. These are images of red blood cells. Not all cells look alike. Some cells have specific functions. Other cells are entire individual organisms of their own. So common misconception, all cells look alike. This is false. So microscopes. We have different types of microscopes. Here we have a compound light microscope. These types of microscopes were what scientists used to make many of the discoveries we know about cells. These are the types of microscopes we'll be using in class. While these are great tools, they are limited and can only be used to see up to 2,000 times what the human eye can see. So that is impressive, but when we get to our next type of microscope, the electron microscope, we'll see why that's not as impressive. We also have electron microscopes. As you can see from the picture, electron microscopes are a little different than compound light microscopes. They use a beam of electrons instead of natural light to magnify images. As a result of this method, electron microscopes can see objects up to 10,000 times beyond what the human eye can see, 10 million times beyond what the human eye can see. So 10 million is far greater than 2,000 with what the light microscope can do. And we can really see high clarity, high resolution images with electron microscopes. So using a light microscope, Robert Hooke developed what's known as cell theory. He observed cork cells and said that living things are made and organized of cells. Additionally, Matthias Schleiden studied botany, the study of plants, and concluded that all plants are made of more than one cell. Here we can see an image of plant cells up close. We will want to remember what plant cells look like for our models of photosynthesis. Plant cells have the characteristic green color because of an extremely important pigment and biomolecule within their cell, chlorophyll. Chlorophyll is what allows plants to absorb sunlight and convert that light into usable energy. Theodor Schwann, another scientist during a similar time as Schleiden, Schwann studied yeast and concluded that yeast, as we will see in our lab, are made of cells as well. Here's an image of our yeast cell. Again, we can see that different cells have different appearances, compositions, and organizations. So we've seen a couple of cells now. We've seen red blood cells, we've seen plant cells, we've seen yeast cells, we've seen a general cell. We can see that they're all a little bit different. Key points to understand, critical points to know of cell theory are these. All organisms, living things, are composed of one or more cells. The cell is the basic unit of structure in organisms, and all cells arise from other cells. A single cell divides to form two identical new cells. So we have two types of cells that are important to know the distinction between. The prokaryotic cells, prokaryotes, and eukaryotic cells, eukaryotes. Below each type of cell, you can see the basic structure of each type. From these images, you can notice that a eukaryotic cell is much more complex in how it's organized in comparison to the prokaryotic cell, which, while has organizational elements, is less complex. Moving along, prokaryotes have no interior organelle structure. Pro means before in Greek, and karyo means nut or kernel in Greek, or derivatives of those words. Breaking down the Greek root of the word, prokaryote means before nut. In contrast, eukaryotes are cells with internal membrane-bound structures. The prefix eu has a Greek root of well or good, so well or good nut. So let's revisit the images. We can see that prokaryote does not have internal organelles and has a loose structure before the nut, while the eukaryote has several membrane-bound organelles within its membrane, much more complex, the well or good nut structure. As we have discussed homeostasis, which is the balance cells must maintain in their internal environment, the plasma membrane is a critical organelle, a boundary between the cell and its external environment. The same way skin is our boundary between our environment and our internal organs, the cell membrane surrounds cells and helps to maintain homeostasis. A cell must maintain a balance in their internal environment. This is homeostasis. And the plasma membrane is a boundary between the cell and its environment. It helps maintain that homeostasis. Structures within the cell are also bound by membranes, and we call them organelles. Organelles are structures within cells that have specific functions in running the cell. And this is a good point to stop in today's screencast, and we will pick back up with another tomorrow or the next day, in which we'll go into the specifics of each one of these organelles. Thank you for watching, and have a good night or a good day.